I'm Daniel Levine, and this is the Bio Report.
One of the biggest hurdles in drug development is targeting proteins found in both healthy and diseased cells without triggering toxic side effects. In cancer, this challenge often translates into narrow therapeutic windows, collateral damage to normal tissue, and forced dose reductions that limit efficacy. The result is a crowded field where many companies chase the same well-known targets, leaving vast patient populations without effective options. Immuto Scientific is taking a different approach. The company is redefining how targets are identified, focusing not on genetic sequence but on disease-specific protein conformations. By studying the structural shapes that proteins take in malignant cells, Immuto aims to distinguish cancerous from healthy tissue, broadening therapeutic windows and unlocking new or previously undruggable targets across oncology and beyond. We spoke to Faraz Choudhury, co-founder and CEO of Immuto Scientific, about the company's AI-enabled structural surfaceomics platform, how it allows drugs to selectively home in on diseased cells while sparing normal ones, and Immuto's plans to extend its science into immunology and inflammation. Faraz, thanks for joining us.
Hi, Danny. Pleasure to be here with you.
We're going to talk about drug toxicity, the need to develop therapies that can target diseased cells without harming healthy tissue, and Immuto Scientific efforts to address this issue. Let's start with toxicity, though. How significant a problem is this in both clinical drug failures and in limiting the benefits of cancer therapies in particular?
Yeah, that's a good place to start, Danny. And, if you look at statistical data, right, toxicity is one of the primary reasons drug fail in clinical trials, right? About 1/3 of all clinical failures are actually due to toxicity and safety concerns. This is even more pronounced problem when you look at oncology because cancer drugs are often designed to be highly potent. They're designed to kill rapidly dividing cells. But most cancer targets overlap with healthy tissues. And that leads to very narrow therapeutic windows for this drug because the difference between an effective dose and a toxic dose can be quite small. So what that means is that a lot of these drugs that entered the clinic, they have significant issues with toxicity, off-target effects, as well as on-target of disease toxicity. So this problem really limits treatment options for patients and really ultimately their survival potential. So if a drug cannot distinguish between a healthy cell and a diseased one, its full therapeutic potential will never be realized.
Why is this such a significant problem?
Yeah, good question. So at its core, this is a targeting problem, right? If you're targeting CD33 or HER2 or EGFR, you're not just targeting the cancer, you're also targeting the protein that is present both in cancer cells and in healthy tissues. So this has two broader effects. The first one is that these therapies damage healthy tissues, right? Because the target is present in healthy tissues. And this can cause some really brutal toxicity and side effects for patients. Patients have ocular toxicity where they lose their vision, lung disease, liver toxicity, kidney failure. So these are really harmful for patients and really affects their quality of life. And to manage toxicity, we often have to start limiting the dosing, right? And as we pull down dosing, we start losing efficacy of these drugs. So we're essentially handicapping our therapies and leaving a lot of therapeutic potential on the table because we cannot distinguish between a cancer cell that we want to kill and a healthy cell that we want to preserve. So the second big problem I wanted to highlight here is that this is really interesting, Danny. So there's a study that came out from McKinsey a couple of years back that on average for a cancer target, there are 9 assets being developed, which means that things are very crowded. Everyone is going after the same few targets that have a favorable therapeutic index. Um, so there's a clear need for new targets, right? Not only because of the competition on targets, but also because a lot of patients are left untreated, right? Because we don't have any therapeutic options for them. So as long as we are defining targets at the sequence level, this toxicity problem is unavoidable. So we really need a new way of distinguishing disease cells from healthy ones.
Immunotherapeutics believes it's possible to distinguish a target protein in a disease cell from the same protein in a healthy cell based on its three-dimensional structure. How is that? What happens to proteins in disease cells?
So we typically think of proteins as static structures, right? Like when we look at a PDB structure, it's a crystallized protein or a vitrified protein for a cryo-EM structure, and it's a static image. But in reality, proteins are quite dynamic. A protein's three-dimensional shape, what we call its conformation, can change based on its cellular environment. So in a disease cell, the environment that the protein is in is fundamentally different from healthy cells. So when you look at cancer cells, for example, they undergo profound changes, right? In the tumor microenvironment, you have stress, glycosylation, mutations, altered trafficking, misfolding. All these stresses can cause conformational changes in protein on the cell surface. And the protein sequence stays the same, but the shape changes, which can expose new epitopes that are normally hidden in the healthy state. So you get a healthy conformation of a protein and then a disease conformation of the protein. And our core insight is that structure provides a pathological signature of the disease, which now we can see for the first time with the platforms that we have developed. We can map those disease-specific conformation, design drugs that selectively bind to the disease version of the protein while leaving the healthy version alone.
You've developed an AI-enabled platform to conduct what you call structural surfaceomics. Can you explain how your platform works? What does it measure that conventional structural biology and proteomics don't capture?
Yeah, so let's start with the conventional methods that you just mentioned, structural biology and proteomics. When we look at the conventional structural biology methods, we are looking at purified proteins, right? The protein is removed from the cell, it's purified, then you crystallize the protein or you do your vitrification for cryo-EM. The purified state of the protein isn't exactly reflective of native biology, right? The native biology of a protein is in a messy, chaotic cell and tissue environment where a lot of different factors are affecting the conformation and the folding of the protein. The other aspect of conventional structural biology is that these methods are typically very slow and low throughput. Uh, Getting a single protein structure can take weeks, months, or sometimes even a year to generate that data. On the other hand, you have your conventional proteomics methods. Those are very high throughput method. You can look at hundreds and thousands of proteins at the same time, and it measures the identity and the abundance of the protein, even in very complex samples, right? So you can look at cell lysates, tissue extracts, but when you're doing conventional proteomics, you're not getting structural data. Our platform is the first to bring structural analysis of proteins to the omic scale. We are combining the study of protein structure in living models of disease with proteomics as the readout. So that allows us to not only understand the abundance and the presence of which protein is in the system, but also how the protein's form or shape or structure is affected in different contexts in a disease cell versus a healthy tissue. And this is where the AI component becomes critical because the data we generate on the structural proteomics platform is very rich, as you can imagine. We're not just looking at one protein in a single state. We're analyzing thousands of proteins across different cellular contexts in various different conformations. So, in order to identify patterns that distinguish between the disease specific conformation and the healthy conformation, we use our AI platform to sort through the data and use a combination of our proprietary structural data and a generative deep learning model to model the conformation in living cells and tissues. So, this is a big breakthrough, right? Because if you look at conventional methods, this was not possible with traditional crystallography and cryo-EM type approaches. Even if you look at some of the new ones like protein folding models like AlphaFold, those models are trained with static crystallized protein structures. So what we are doing is we are creating a map of the disease proteome that goes beyond just protein expression levels. We're capturing the structural dimension of disease biology, and that's a whole new drug target space that is invisible to today's platforms.
Well, walk me through the process. How do you go from a target to a therapeutic candidate?
Yes, this is a very streamlined process that we use, right? So we start with patient-derived disease models. These samples come from our academic partners. We're partnered with three of the top academic medical centers in the US, the University of Wisconsin, University of Pennsylvania, and Mass General Hospitals. Once we get the samples, we run it through our rover platform to identify and model proteins that demonstrate dramatic structural changes in the disease state. And then we use our internal large language model to prioritize and triage this list of targets. As you can imagine, when we run these analysis, we actually see dozens and sometimes even hundreds of proteins that are uniquely structured in the disease form. So we need a way to sort this and rank them. So we use several factors in our LLM to rank them. We look into the degree of structural change, how stable and persistent these changes are across multiple different samples, the relative expression of the protein in both the disease-specific expression as well as in the plasma membrane, as well as the literature, right? Is there any known biology about this target and its association with the specific disease we're interested in? So we generate a rank list of high-quality therapeutic targets with a weighted scoring system. And then from that list, we would pick the target of interest. Sometimes it's more than one target, but we pick the one that we would want to go into the antibody design stage. Um, the first part of the antibody design is identifying the disease-specific epitope. And then we use our radar platform to essentially start designing an antibody. The radar platform is a AI stack for antibody engineering. And what it does is that it takes into the structural data that we generate on the epitope and the conformation of the protein to generate and design binders against those specific epitope, right? We do this on a residue level, which we know exactly which amino acids we want to hit. And we're able to do that pretty quickly. And then we validate that in the lab. We generate the antibodies and we do the screening in-house to make sure that the antibodies are binding to the right epitope. So that's the antibody generation part. Once we have the antibody, we then do a very thorough target validation. And how that works is that we use the antibody to screen both fresh and frozen archival patient samples. We can screen large cohorts. Oftentimes we do 50 to 100 samples to really determine the prevalence of the target directly in patient samples, right? And this is a crucial step because these are novel targets. We need to validate the targets directly in patient samples instead of relying on, let's say, in vitro assays in cell lines, which may not be a very good representation of the disease biology in real patients, right? So we do the screening for validation of the target. We also screen samples from healthy donors to ensure the target is not present in healthy tissues. And that gives us a lot of good information that we need to kind of take into account to select the final target that we want to move forward with. So then now we have the target, we have the antibody. We then combine this antibody with the right modality. For example, ADCs are quite common modality that we use. Because it's a disease-specific structure, we can use that disease-specific structure as an address to deliver a cytotoxic payload to tumor cells. We also work with bispecifics and multispecifics, T cell engagers, for example, quite often. So essentially, it's a pretty tight loop. We identify unique structural signatures, design binders to that signature, and then configure the modality based on biology.
How specific are the therapeutics you're able to create to target a protein in a disease cell? And you mentioned a couple of different modalities, but they're essentially antibodies that you're using. Is this approach able to use other modalities other than antibodies?
Yeah, great question, Danny. I'll take it in two parts. First is on the specificity. The specificity we have achieved is actually quite remarkable. We see selectivity ratios of 1,000 to 1 or greater. And that kind of selectivity really translates to a much wider therapeutic window than what we see on other drug programs in the clinic. And that's really the goal, right, is the reduced toxicity profile. Um, in terms of our modality, you're right, our platform is largely agnostic. The choice of modality really depends on the specific target, the binding site characteristics, as well as the therapeutic goal. So even though the majority of our work today has been with ADCs, but these targets are actually very well suited for modalities like bispecifics, T cell engagers, or even some of the peptide and small molecule based approaches.
Is there a potential to rescue therapies that were previously considered too toxic by altering the sites at which they bind to a protein?
Yeah, you're absolutely right there, Danny. So a lot of failed programs, right? They weren't bad ideas. They hit the wrong epitope, right? That caused toxicity in the clinic. We can design a molecule that binds to specific, very disease-specific epitopes because that's the thing that we identify in the first place. And if we can do that, we can really reduce some of the toxicities of these programs and salvage this otherwise promising biology. So there's really a long tail of failed assets that we think could be redeemed using the approach we're taking.
Does this at the same time open the potential to hit targets that were previously considered undruggable?
Yeah, so undruggable is really an interesting term, right? There are many reasons why a protein may be considered undruggable. The most common one is that the undruggable protein does not have a clear binding pocket. And a lot of these proteins are actually very challenging proteins to analyze, right? You cannot crystallize them. They're multi-pass membrane proteins. So if you try to purify them, it's hard to do so. Our platform actually does not have that limitation, right? We can work with any protein because we do the analysis in living cells without having to purify these proteins. And a lot of the clients and partners we worked with, they came to us with proteins. They did not have any structure, right? They didn't know if there's a binding pocket that they can go after. But we were able to characterize and really define the structure of that protein, and oftentimes find potential binding pockets, right, where they can target. The other aspect of that is that because we look at the protein in both its disease conformation and its healthy conformation, oftentimes we see that even though there might not be a clear binding pocket in the healthy state or the crystallized state, if you look at the same protein in the disease state, we see new pockets opening up or new epitopes showing. So that really leads to opportunities of targeting some of these undruggable proteins.
I suspect there's an economic case to be made here for Immuto's approach, given that the high failure rate of drug development contributes significantly to its high cost. Is there anything known yet about the speed, cost savings, or efficiency of your approach?
So we're still early in terms of our pipeline development, right? But we're seeing two patterns that are emerging. The first one is that better target selection upfront can avoid a lot of the costly attrition that happens downstream, especially in late stage clinical trials, right? And that's the big thing that we're really focused on is can we pick better targets that are more disease specific so you're not taking the risk in the clinic, right? When you're picking a target that is highly expressed in tumor cells, but it's also expressed in healthy tissues, you know that there will be some toxicity when you're going into patients. You're just kind of placing a bet that the toxicity would be manageable. But because we are targeting proteins that are highly disease specific, we think that we can really cut down on a lot of the attrition that happens in clinical trials. So that's the first thing. The second thing is that the hit to lead validation of our antibody discovery process is actually much faster than some of the traditional methods because we are structurally defining the epitope, as well as we are using our modeling system to design the antibody against those specific residues. So this can go much faster than your traditional antibody discovery methods. But I'd like to point out that even though this is a streamlined process, saving a few months in discovery is really not the big bottleneck in drug discovery, right? It's that drug development is expensive because most projects fail. And if we can reduce failure rates in clinical trials by picking better targets, getting better selectivity, that would really make the big difference, reduce costs, reduce timelines, and hopefully get better treatments for patients.
Your initial focus is on cancer, although I suspect the platform could be applied to a broad range of diseases. Why focus initially on cancer?
Yeah, that's a good question. So cancer, it's a disease area where toxicity is a big limiting factor, right? So a lot of the cancer therapeutics have very narrow therapeutic window, and we think that the approach we're taking will make a immediate impact in cancer. At the same time, it's a disease with high unmet need, right? We all know that cancer, not only it's deadly, it's actually ranked the second leading cause of death worldwide. We made a lot of advancements in oncology, but it's still the second leading cause of death for patients. So, we think there's a big opportunity both commercially and in terms of unmet need. It's also a system where conformational stress biology is extreme because, as we all know, tumor microenvironments are pretty divergent compared to healthy cells, right? Um, but that said, you're right that the platform is broadly applicable. We're interested in other indications as well. For example, in immunology and inflammation, where dysregulated immune cell proteins adopt disease specific conformations. So we're very much interested in I&I, as well as things like neurodegenerative diseases, right, where protein misfolding is central to pathology. So we're starting with cancer, but our vision is much broader.
Your lead experimental therapy, IMTO 4842, is in development to treat acute myeloid leukemia. What is IMTO 4842 and how does it work?
Yeah, so IMTO 4842 is an ADC. It's very specific for a structural target on AML tumor cells. The healthy protein is present on immune cells, but the disease conformation is uniquely exposed on AML blasts. So the ADC that we're developing, it only binds to the AML specific conformation and does not bind to off-target cells such as healthy progenitor cells or hematopoietic stem cells in your bone marrow. So this allows us to selectively kill leukemic cells while sparing healthy tissues. So for the ADC, we selected a stable linker technology as well as a clinically validated payload. So that combination gives us a lot of confidence that when we enter the clinic, we will have a good safety profile and good efficacy in patients. And the indication is that when we look at preclinical models with our drug, we're seeing very potent anti-leukemic activity, right? We have a very differentiated safety profile as well compared to molecules that target CD33 and CD123, which are the more common AML targets right now. And those targets are present in healthy progenitor cells. So, given the specificity of our target, we're very excited about the data. We have achieved the disease selectivity that we're looking for, at least in our preclinical studies. So we are really excited to move this program forward.
And why start with AML?
Yeah, great question. We've talked about this internally a lot, right, as we started building our internal pipeline. And what we the consensus was that our company's goal is really to help patients that currently do not have any therapeutic options. So, as an industry, we've made a lot of progress in oncology. We made a lot of advancements, but there's still a lot of patients that are left untreated, right? Because there's no therapeutic options for them. So, our goal as a company is to target those patients who currently do not have any options. And really, those are the most challenging types of cancers. And those are the cancers that we're going after. So, AML is actually a prime example of a challenging cancer, right? It's very aggressive, it's in their blood and bone marrow. Over 100,000 patients per year are diagnosed with AML, and unfortunately, they succumb from the disease eventually. um, the current therapies, there are some targeted therapies for AML that are specific against specific genetic mutations of the disease. But all these targeted patients will eventually relapse because none of the targeted therapies are curative. And once they relapse, there's really no uh, treatment options for a lot of these patients. So we're developing a therapy that can treat all refractory and relapsed AML patients. And the approach we're taking with the structural surfaceomics and going after a highly AML specific target, we hope that this will allow us to make sure that the patients, AML patients who are already very sick, right, that they do not have to suffer from some of the severe toxicity that current therapies often caused. So we assembled a world-class team to do this with some of the top leading AML experts, including clinicians, researchers, translational scientists who have been working on AML therapeutic development and treating AML patients for several decades. Additionally, we saw that there's a clear regulatory path for AML, which is pretty well established. The FDA has been very supportive of novel AML mechanisms, as well as the history for accelerated approvals based on response rates in refractory patients have been pretty positive. So this creates a potential faster path to proof of concept and eventually getting to patients as quickly as possible.
Well, what is the development path forward?
Yeah, so right now we are conducting NHP tox studies with our lead program. At the same time, we're initiating our antibody and ADC manufacturing process. We will be establishing a regulatory team to obtain our IND in 2027. And we are also putting together and assembling our clinical development team to initiate clinical trials and towards the end of 2027.
The company has discussed expanding beyond cancer into immunology and inflammation. How will you decide which indications you'll pursue?
Yeah, so immunology and indication is really interesting to us because the same biology of structural dysregulation under cellular stress happens in multiple different areas of I&I, right? Autoimmunity, fibrosis, chronic inflammation. So our goal is to really focus on areas where we expect to see strong protein structural changes in the disease context. And also, we also look at the commercial value of the disease area, right? So I&I obviously uh, is of interest in terms of the commercial value there. So what we have done in the I&I space is actually we partnered with a large pharmaceutical company on a specific I&I indication. We haven't made any public announcement yet, but there will be a press release coming out in the next few weeks. So you can keep an eye out for that. But our goal is to take really a portfolio approach, right? We want to build a pipeline that demonstrates the breadth of the platform while focusing on resources where we can have the most impact.
Immuto started out by providing analytical research and CRO like services to pharma and biotech partners. How has your business model evolved from pure services toward an integrated model that includes internal therapeutic programs?
Yeah. So as you mentioned, we started as an analytical partner to pharma, and a few reasons for that, right? So one is that it funded some of the early platform development work that we did internally. And another important reason was that it allowed us to build relationships in pharma. So when we did some of the analytical partnership work with pharma companies, we've worked with uh, 8 of the top 10 pharma companies globally, right? So it allowed us to really work with a lot of the internal people in these pharma companies. They get to see the platform, the data that we provide, build a relationship. So once we start talking to them about deeper partnerships, right, for target discovery and developing antibodies and so on. They already know us, they know the platform. So the platform is validated through some of the work that we have done together. And it really enabled us to jump straight into some of these partnerships without doing a lot of these kind of validation studies or pilot studies on the front end. But at the same time, from day one, the vision has always been to be a therapeutics company that is powered by the proprietary data sets that we have, right? As I mentioned, we are sitting on a lot of really valuable data of proteins and their structures and conformations in different cell lines and in different diseases. So our business model now is really a hybrid. We have an internal pipeline. We are doing data partnerships with different pharmaceutical companies where they can get access to our data to train internal models. We're also doing discovery and co-development partnerships. So this type of an integrated model really gives us optionality, right? We can advance our own pipeline programs. We can partner them at various stages. And we can also do some of these partnerships that can support platform development while building out our pipeline.
And how do you think about balancing those two, the revenue generated through the service side versus the value creation in your own therapeutics, and do you expect that mix to change over time?
Sure. So platform revenue, that helps us extend our runway and de-risk some of the early financing stages. But really, the long-term value sits in the internal pipeline, right? Over time, we think that there's going to be a shift towards the co-development and the internal asset development model as more proof points emerge.
You recently announced a discovery collaboration with Daiichi Sankyo. What does an ideal strategic partnership look like for Immuto? And do you see Immuto primarily as a discovery engine for large pharmas, or do you envision taking select programs deep into development on your own?
Sure. So for our internal programs, we plan to do the early clinical development work, and then we'll partner with another company who has a scale in late development and commercialization. So that's our strategy for some of the internal pipeline programs that we're advancing. At the same time, we will continue partnering on the platform and early discovery, right? Because if you look at our platform, right, it's an engine for new biology that can really power multiple large pipelines. So, from that perspective, our goal is to partner with pharma companies in orthogonal indications that will really allow us to really maximize the impact of our platform. And ultimately, we want to do this for patients that are missing out on possible cures, right? Which often means that we have to tackle their disease from different angles. And we want to do this both independently and with our skilled and experienced partners that we have.
Immuno's raised multiple seed rounds, including a recent 8 million seed two financing. What's been the biggest challenges in convincing investors that deep structural proteomics and surfaceomics can be a scalable business, not just an interesting technology? And how have you addressed those concerns?
Sure. So I think that's where some of the early analytical work we have done with multiple pharma companies helped quite a bit. Not only it validated the platform, it also showed our investor that there's a big need for it, and our pharma partners are interested to work with us and get access to our platform. But at the same time, we have been very fortunate that when we went out to raise our current round, which was the $8 million seed round that you just mentioned, there was a lot of interest from investors because where we are as a company, we initially targeted a $5 million seed round, but looking at all the interest we had, we extended to $8 million, and even at $8 million, the round was oversubscribed. And I think it's really because we have validated the platform, we are generating revenue. The company was profitable the year before from the revenue generating activities. So really, the pitch was that we're not fundraising to extend our runway. The financing will allow us to really uh, go after some of the high value targets and pipeline programs that we're developing, and to do it with speed and scale.
And how far will existing funding take you? And what's the plan for raising additional capital?
Yeah. As I mentioned, given the revenue that the company is generating, we think about runway a little differently. Where the company has quite a lot of runway, we're really focused on hitting our next milestones, the big milestones that we have. With the current funding, the big milestone is that we want to complete all the in vivo studies. We want to get NHP tox data for internal programs. And then we're planning to raise a Series A round. The goal is sometime next year, we're going to raise our Series A, and that would fund our clinical trials, phase 1 trial for at least 2 of our internal programs.
Faraz Choudhury, co-founder and CEO of Immuto Scientific. Faraz, thanks so much for your time today.
Thank you, Danny. It was a pleasure to talk to you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.